Let's go ahead and get started. So thanks everyone for coming to this in the latest installment of the NMDC Community Conversation Series. Great to see some familiar faces in the Zoom attendees and some new names as well. So thanks all for attending. So if you're not familiar with the NMDC, so the NMDC is a new initiative that aims to empower microbiome researchers to be able to access and truly leverage a lot of the diverse data pertaining to microbiomes, whether it's genomics, transcriptomics, metabolomics, or environmental data, which requires a lot in the area of metadata standards and ontologies, which is what we're going to be talking about today. So this is part of the NMDC Community Conversation Series. So this is some monthly webinars that we hold to really engage with the broader microbiome community to help kind of like, help it kind of explain some of the breadth and depth of the NMDC program and engage in some like conversations with you and kind of also give an opportunity to help, shape the future directions of the NMDC. So your feedback is really welcome, and thank you all for attending. So just to give you an outline of how it's going to work today. So I will start by just giving a brief introduction to ontologies in the context of microbiome research for about 10 minutes or so. And then we're lucky to be joined by three panelists with very diverse expertise. Blumberg from Bicodemo, Ramona Walls from the Critical Path Institute, and Tom Sharpton from Oregon State University. They'll kind of give you a bit of an intro to their work. And then after that, we'll have a panel question and answer session. For this, we have some shared notes. So you should see the link in the chat. Marissa, I don't know if you want to put that there again for new attendees. So feel free to put your questions down there. Also, if you like, you can also put your name and affiliation there. And also want to point you at the code of conduct at the top of that documentation. Like having an NMDC, we abide by this code of conduct. So with that said, let's go ahead and get started. So just to kind of like set the scene a little bit, I just want to kind of like mention how ontologies are being used in some other areas of the life sciences. So I'm also part of the Gene Ontology project. So I think the Gene Ontology, or GO, should be familiar to many of you doing genomics research. What GO gives you is a set of standardized terms that can be used to annotate the functions of genes in a variety of organisms, either manually or automatically. And it's used for a variety of purposes. You can make use of these annotations to build gene sets. And maybe like one key killer application of the GO is gene set enrichment, which is how many of you will have encountered GO. You'll have set of genes that are overexpressed under some experimental condition, and you want to understand what distinguishes those genes from other genes in the genome. So so that's one real kind of like key application of the GO. Another example of another ontology is the human phenotype ontology, which can be used to annotate the signs, symptoms, and phenotypes of individual patients with diseases, or to kind of characterize diseases more broadly. So, a killer app for the human phenotype ontology has been as part of kind of like new genomic medicine initiatives, where it's actually being used to kind of like diagnose patients with rare diseases. So there are many applications, many different ontologies with many different applications, but I think it's helpful to think of some of these kind of key applications. So why, that's all very well, but why might we want ontologies for microbiome research? So I thought I'd frame this in terms of maybe a kind of a research question where a researcher may be interested in finding, broadly, a set of microbiome data sets. They may come from different databases, genomics databases, metabolomics databases. with maybe some kind of characteristic in common. Maybe they're interested in wetland biomes, and they may also be interested in how different environmental characteristics of the microbial community, such as things like peat death, might affect the composition and function of these microbial communities. And at the moment, to be able to do this, it can be quite challenging. You need a kind of shared vocabulary to be able to kind of query across these different databases. If in fact the databases actually store sufficient metadata to allow you to do this query in the first place, often it may be the case that this isn't actually captured. So there's really no way to kind of like go back and do this kind of meta-analysis. So what ontologies give us is a standard shared vocabulary for being able to query with both within and across these different databases. So one of the key ontologies here is EnvO, the environment ontology. And I can show you just how this fits in within a kind of like a broader framework here. EnvO really works in concert with MIXS, the minimal information about any sequence standard from the Genomic Standards Consortium. MIXS gives you a standard set of terms that can be used essentially as a data dictionary to describe the columns in your data set. So there may be many different kind of quantitative and qualitative parameters in your data set, the environment in which a sample is captured, as well as kind of like measurements such as salinity, metal content, the depth of the soil sample. So all of these kind of column headings, can be standardized using MIXS. And for the actual qualitative values themselves, we can make use of ontologies such as EnvO to be able to standardize these so that we have like, one standard term for describing a wetland or shore or sediment versus soil and so on. EnvO works in concert with other ontologies. So, for other parts, maybe if you're looking at samples that come from an experimental study, you may want to be able to query by the experimental treatments and so on. And this is where other ontologies like PICO, the plant environment conditions ontology, would come in. So these ontologies all work in concert, and in fact, many of them are part of an initiative called OBO, the Open Biontologies Foundry, which aims to provide a set of well-integrated modular ontologies, plus the framework for being able to link all of these ontologies together to be able to annotate your data, whether it's genomic data, whether it's microbiome sample data, and so on. So some examples of OBO ontologies are EnvO, PO, the plant anatomy ontology. For human microbiome samples, you may be interested in describing the sites on the human or metazoan body from which the sample was captured. That's where an ontology like Ubron would come in. PICO for plant conditions, GO for describing function, and so on. Having said that, we need to acknowledge that ontologies, can be challenging for people to use. They can often be, kind of quite opaque and hard to kind of like, understand exactly how to use them, either for annotating your data or for querying the data. So one of the questions we're interested in exploring here is how can we make it easier for submitters to annotate using the relevant terms, as well as make it easier for people to be able to query data using these ontologies. And we also have the challenge of multiple different overlapping systems for being able to describe aspects of samples, whether it's overlapping systems like kind of EnvO and EMPO for describing the environmental context, as well as multiple different systems for being able to describe experimental factors such as treatments and so on. One thing that we are doing to address this within the context of the NMDC is we are actually creating subsets of these ontologies such as EnvO, and these are context-specific subsets, such that if you have, for example, a terrestrial soil sample, then we will provide you with a simple subset of EnvO that for each of the different kind of like key fields within MIXS. So this is shown here in a new application called Data Harmonizer, which is a user interface that actually comes from the University of British Columbia, but we're working with this group within NMDC to kind of like tune it towards kind of like a microbiome sample standards. So you can see here, this UI allows you to kind of like, it provides a more user-friendly way for users to enter their sample data and to be able to kind of like access some of these ontologies without being an ontology expert. And we're also aiming to make things easier for people to query data using ontologies through the NMDC data portal. This screenshot shows an example of our kind of ontology-based faceted view. The user here may not even be cognizant that they are using an ontology here, and hopefully things just work. Leveraging the ontology in such a way that they get the right results for their kind of like questions at hand. So, for example, if they're interested in terrestrial biomes and they select that, then it should also include using the hierarchy in the ontology, different kinds of terrestrial biomes such as forest ecosystems and so on. I invite you to also try this this new user interface as well. Okay, so with that, I think we're on time here. So I want to just kind of invite the panelists, and please, if you have questions, just put them in the Zoom chat or in the Google Doc, and we'll get to them later. But before taking those, I want to hand it over to our panelists in the order here. So why don't we get started? I'll stop sharing, and you can introduce yourself, Kai.
Great, thank you so much, Chris. Go ahead and just share. Let me know if you can hear me. Great. Okay. All right, and I assume you can see my screen as well.
Yes.
All right, great, thank you, Chris. All right, thank you again, everyone, for the invitation to be here. Got an interesting panel, so I will try to keep into the 4-minute time frame. My name is Kai Blumberg. I'm a PhD candidate at the University of Arizona, and I'm also an ontologist at the Woods Hole Oceanographic Institute's Biological and Chemical Data Management Oceanographic Data Management Office, BICODEMO. It's quite a mouthful. Okay, so just really briefly, just some relevant work that I've been working on in my job and my PhD, which I think might be helpful to this discussion. Just a few things. So in terms of ontology development, I help contribute to the environment ontology, which Chris mentioned. I've also worked on several other OBO projects, including some work to help make units a little more interoperable. I could talk about that, but we don't necessarily have to. And then more work about NMDC relevant work that I've been up to, I will be presenting on really briefly, is the BICODEMO semantic model, and some work in my PhD with Planet Microbe. So I know Chris mentioned this, so I'll keep it really brief. The OBO Foundry has this cross-disciplinary set of interoperable ontologies. In theory, at least, and mostly in practice, they work together in a way that machines can understand what each other mean in a common way that's queryable to humans, but also to machine systems, so that when we set up our systems to ask for data, we can search this large web of knowledge to actually find things that are relevant and also have been contributed to by thousands of people across the world. Yeah, so he mentioned a few of these. There's a few more. It's all a big family. We can talk more about this later. There are many ontologies that are also not part of the OBO Foundry, just to keep that in mind, but the people here are more OBO people, so the discussion will probably be more in that line. For example, the environment ontology, which again he mentioned, terms to cover many different domains: microbial ecology, oceanography, cryospheric sciences, hazards, pollution, soil sciences, manufactured products, perhaps a few too many things. And of course, this is used within the MIX-S standards to kind of help bring that minimum metadata standard into at least some sort of actionability, in that people, when they annotate their data, they put, the broad scale context of their sample and using an EnvO term, or the local scale, or the material. So just turning briefly to some of the work that I'm doing at BICODEMO, just really briefly. BICODEMO, the Biological and Chemical Data Oceanography Management Office, is basically where the NSF oceanographic cruise data is supposed to go. Doesn't always, but that's in theory what's supposed to happen. And so we had the opportunity to kind of revamp their system. And again, there's a lot of nuances in the marine world. The NERC vocabulary server is quite big, but we felt that wasn't quite right for BICODEMO's purposes. So what we ended up doing was kind of making a hybrid model between kind of the semantic model that's put together like the NERC vocabulary server, but I pushed really hard to have the OboFoundry ontologies be the vocabulary terms that actually get integrated in to the new BICO-DEMO semantic model in order to make this forward compatible and interoperable with systems like NMDC and others. If we can all be using the same vocabularies, at least we're laying the groundwork for our data to be interoperable. I think that's really important with this, this oceanographic data to be able to communicate, or at least be in the same vocabulary, to be able to communicate in the future with things like the NMDC. Yeah, so just a little bit of the model phrase. It's very similar in terms of making subsets. So for example, in the matrix field, I basically tried to copy the mix-S standard and bring this right into the BICO-DEMO model so that it would be, at least in that way, interoperable with NMDC and INSDC data. And then similarly to what Chris just presented, made subsets for the different submodules for our annotators, the BICO-DEMO data curators, to be able to, more comprehensively annotate things using terms from, these OBO ontologies, which we've been talking about. And then really briefly, my PhD work, Planet Microbe, it's kind of like a precursor to NMDC, very similar scope, but much smaller project where we just focused on marine data. And basically we've come up with some of the similar things to NMDC, but, we're just like have a little bit further ahead of a system, but it's also not, continually supported. So it's kind of a one attempt at what something like NMDC, which has longer term funding, could become. And so the long and short of it was that we kind of found a way to containerize data, again, making use of ontologies from the OboFoundry to express, things like what it is, like the concentration of chlorophyll in liquid water, in what units, and how it was measured using a device from another ontology, the Open Biological or the Biomedical Investigations ontology. And all that, comes together within a portal where, things are made searchable by, their EnvO contextual terms, as well as qualitative, quantitative terms, such as concentrations, et cetera, so that it can all be searched and queried. And finally, kind of the tail end of this is that, as Chris mentioned, using the gene ontology is one way to explore a bit further into our microbiome data in terms of the function of, what's there. The NCBI taxonomy ontology, which is basically just the NCBI taxonomy database expressed in an ontology format, is also a good way, like a hierarchically structured way of, getting at the function of our data. So in the last stages of my PhD work, I'm working on some analyses where we can actually use ontologies to drive analyses about our data, specifically with marine sets, but this applies more generally. So basically the idea being that we can use the knowledge and information that's captured within ontologies like Go and CBI Taxon and EnvO to search the machine for relevant information pertinent to a question, and then just subsample that information and deliver that to an analysis, like a statistical analysis. So we go from a lot of data to a subset of useful data, search like anything that's in the sub tree of ATP generation, et cetera, processes, and then get back that data. And so that allows us to ask questions like what cellular, sorry, I used a different example before, but what cellular amino acid biosynthesis processes differentiate sediment from different marine benthic biomes. And, so we go from very complicated genomic information, subsample using the gene ontology, and get back a smaller set of terms that we can actually analyze and maybe see what the differentia are. And here you can see just a brief example from my master's thesis where I set up a prototype system to do this, and you can see that the bathol and the neritic samples did ordinate differently, and then you can see there are gene ontology terms more commonly found, more abundantly found in some rather than the other, like asparagine biosynthesis in bathol and cysteine biosynthesis in neritic. Yeah, I hope I'm not too over time. Thank you so much, and I'm happy to take questions afterward.
Thank you, Kai. Let's move on to Ramona.
Hi, everyone. While I get my slides loaded, my name is Ramona Walls. I am the Associate Director of Data Science at the Critical Path Institute, and it turns out I can't do two things at once. I was trying to introduce myself and do the slides at the same time, but it's just not good. So the Critical Path Institute is a nonprofit organization located in Tucson, Arizona, with the mission of accelerating the development of treatments for diseases, particularly in underdeserved and rare diseases. I also have an appointment at the University of Arizona, where I'm assistant research professor and manage a couple of small grants there. So just to give you a sort of an overview of what my ontology experiences are. Up until my taking the position at CPATH, I primarily worked in biodiversity related ontologies. So I'm the lead developer for the biocollections ontology and for the population and community ontology. So yes, I'm the person you complain to when you requested a new return in those ontologies, and the release has not come out yet. Please bug me if you need that. The plant ontology, that's how I got started in ontologies, was as a postdoc developing the plant ontology, which should be PO, CO. I occasionally contribute to EnvO and more often instigate EnvO on things that I would like to see change there. Kai and I work together on the Superfund research project Data Integration Ontology, which is an environmental health ontology that brings together data from many different sources, including both stressors and resiliencies. So it's got things like exposures to chemical, poverty level, but also whether you have access to medical care, education, et cetera. So this real interesting combination of environmental factors, both like hardcore, like physical environment factors and social determinants of health as environmental factors. I've worked on a number of different application ontologies that use the BCO model to integrate data. And currently in my position at the Critical Path Institute, we're developing something called the Rare Disease Cures Accelerator Data and Analytics Platform, or RDCA DAP. It just launched in September with 59 data sets across 23 different rare disease areas. We are using various terminologies and ontologies in there, including OMOP, CDIS, SDTM, FIRE, and on many of the OVO ontologies as well. So just to give you an idea of the kinds of challenges and problems that I solve with ontology. So in addition to building domain and application ontologies, what I really focus on in my work is building workflows and applications and tooling to integrate data from diverse sources using ontologies. So this is an example from a fairly old paper now about what one of the key use cases of the biocollections ontology, which is to track the provenance of samples and data through different processing chains. I've also done quite a bit of work on aligning observation ontologies. So don't worry about trying to read this slide. It's just showing that we had an effort where we got together with about 4 different semantic web ontologies where we were looking at how can we model observation data. These are data where someone goes out and takes a measurement of, the temperature of seawater or the height of a plant, et cetera. And so we managed to achieve this core model that aligns across these many different ontologies, including OVO Foundry ontologies. And finally, I'm going to end with something that's not so specifically ontologies, but is really relevant because through my work on developing and using ontologies, I realized that we couldn't do a lot of the work that we needed to do without the use of good identifiers. We need globally unique and permanent identifiers for physical specimens, for data sets, and for various other objects. So I've got 2 active grants right now. One is called iSamples, and one is called Sampling Natures. iSamples is an NSF infrastructure grant where we're building a domain neutral set of services to serve for minting and minting identifiers and recording metadata about material samples in the life history domain. And Sampling Nature is essentially a research coordination network that's going to help address many of those social issues around globally unique identifiers for samples and sample-based research. And so that's just a quick overview of myself, and thanks. On to the next person.
Thanks, Ramona.
Sure.
Let's move on to Tom.
Well, good morning, everybody. It's a pleasure to be here. Let me just load my slides for you all. All right. So by way of introduction, I'm Tom Sharpton. I'm an associate professor in the departments of microbiology and statistics at Oregon State University. Now, I should note that I am by no means an ontology expert, but I am someone who has used ontologies to solve problems in microbiome data science in my lab's various research investigations. And what I'd like to spend some time talking to you about today is at a very high level, the types of questions we're interested in, how we study them, and provide some context for how ontologies have been or could be useful for the types of research that we're conducting. So my lab seeks to understand how the gut microbiome contributes to homeostasis and health, and how perturbations to the gut microbiome yields disease and disorders. To address this question, we tend to apply a variety of systems biology research tools, wherein we characterize the various features of the microbiome and its environment, which in the case of our work would include the host. And we apply then computational and statistical approaches to resolve associations between these different features, maybe microbes and pH, for example. As these types of associations could indicate potential linkages between these different types of features and help us uncover mechanisms of interaction between them. So our efforts might, for example, include using shotgun metagenomic sequencing to characterize the genomic composition of the microbiome, computational tools that then identify and functionally annotate the genes represented in the metagenome, and statistical approaches that reveal associations between specific genes and maybe the host's health state. By way of example, I'll introduce you to some work that we did a couple years ago, where we obtained a litany of publicly available data sets from a variety of human gut metagenome clinical investigations. All right. And we were interested in doing a meta-analysis on these data to identify gene families that robustly associate with different human diseases across studies, as well as gene families encoded in the metagenome that robustly associate with disease across different diseases, all right? And so to do so, we analyzed each study participant's metagenomic sequencing data and quantified the relative abundance of each detected protein family in that sample. And we then compared these protein family profiles across samples and used regression approaches to resolve specific protein families that indicate a specific disease. We call such protein families disease indicators. They indicate a disease. All right. Well, what I really want to highlight coming out of this study is the result of our comparison of these diseases across the different types of diseases that we considered. All right. We looked at a variety of different clinical studies that were focusing on different human diseases and disorders. And what we found when we looked at the disease indicators resolved in each of these different diseases and disorders can be illustrated in this chord diagram. Here on this plot, you see on the outer ring the different indicators that were identified for a given disease, like type 2 diabetes. All right. And what we ended up doing is we then assessed whether or not an indicator in one disease was also revealed to be an indicator in another disease. And these arcs in this chord diagram illustrate such instances where we revealed or identified indicators of multiple diseases. Now, this is an interesting observation that has uniquely come out of this type of a data integration approach, because it indicates that these types of protein families may link to health or disease in a general context. Maybe because they're responding to similar types of physiological perturbations like inflammation, or possibly because changes to the relative abundance of these types of gene families breaks down the microbiome's contribution to homeostasis and allows different types of diseases and disorders to emerge. So I'm mentioning all of this not just to illustrate the power of data integration approaches, but because I think that one of the places where we see ontologies playing a really critical role is in the integration of diverse data sets, especially from publicly available databases. I think that ontologies can play a profound, provide a profound service to the community in the sense that they allow us to appropriately identify and integrate the right type of relevant data into such studies. We need to know if the samples that we're looking at, for example, are relevant to the types of comparisons that we intend to conduct in our investigations. And having some shared language about what the samples were derived from not only allows us to search through publicly available databases to find relevant samples and data, but also to help ensure that the data that we're ultimately integrating is meaningful in the study that we're conducting. Moreover, ontologies allow us to improve the interpretation of microbiome data by providing some knowledge context about features that we uncover and discover in our statistical investigations. Let's say, for example, that I identify a microbe that links to some feature of interest. Well, now I've got to try to figure out what I happen to know about that microbe. And perhaps ontologies can ultimately be developed that allow us to determine the potential functional or ecological roles that are known about those microbes to see if that improves my ability to infer meaningful hypotheses hypotheses about the informatic discoveries that I uncover in my investigations. So that's what I wanted to cover at the outset. And I suspect that we'll dive deeper into these ideas through today's discussion about this interesting and impactful topic. Thanks so much for having me here today.
All right. Thank you, Tom, and thank you, everyone. And if the panelists want to put on their cameras, and then we can move to the panel discussion. So I'll get us started with a few questions. So we heard some kind of like, great examples of some applications there. I wonder if we could kind of dive deeper and just, if all of the panelists could kind of give their perspective on, if there was like one kind of like killer app or one like really, what is the one most impactful, area in which kind of like using an ontology or using a set of ontologies could impact microbiome research, and which ontologies would those be? How would that work? So I don't know who wants to take that first. Or...
I'll dive in. I'll get things started at least. So I think the limiting factor is getting data annotated with ontologies, right? So there's always work to be done on the ontologies, but it's getting data into a well-curated form that's tagged with ontology terms. So I think the killer app would be on the user-facing side, on the researcher side, so that when they're creating their own data, they can tag it easily with ontology terms. What that would look like could be a much longer discussion.
Yeah. Mm-hmm. Right. Kai? And
yeah, just briefly, I think in the future, data that is not annotated is not going to be useful to the collective in a meaningful, like discoverable Star Trek computer kind of way. So I think whatever we can do to get more data annotated and have appropriate sources of information to annotate that data and get that into some sort of centralized hub, or at least set of formats, or at least set of protocols that allow for kind of decentralized access to federated queries across all the different data spaces that we require to make our killer apps, because like all the statistical tools and applications, that all depends on the input data sources. So agree with Ramona on that point.
I mean, I certainly agree, as someone who's perusing public catalogs of data to integrate into our investigations all the time. It's very challenging sometimes to obtain ontological annotations of the data that we seek to integrate. And that's, from our perspective, challenging and sometimes frustrating. And so there's tremendous need there. To maybe take the conversation into a different direction, I'd say from an application standpoint, something that would tremendously help the field are ontologies that improve our ability to interpret the results that come out of our statistical investigations. It is a common problem in microbiome research for somebody to say, look, this amplicon sequence variant in my study can be taxonomically annotated into the Bacteroides, and it is linked to some study outcome in my investigation. What do we know about this particular group's relationship in other investigations? And an ontological framework that affords us the ability to computationally peruse through that domain of knowledge could transform the rapidity with which types of discoveries are resolved in these types of investigations. I know that that OMI, the ontology of host microbe interactions, is one example of a framework that is attempting to do this, and to leverage publicly available discoveries and encode them in a way that allows investigators to rapidly assess other types of associations that have been uncovered for certain kinds of organisms. And I think these types of frameworks would fundamentally transform the types of discoveries that can come out of microbiome research.
Yeah, I wonder if you can say a bit more on just what some of those terms might look like. It sounds like some of this would be terms to describe the gene families themselves. So maybe standards like PFAM and Interpro. Some would be standards to describe the function, some to describe the taxonomy, and some to describe, I guess, the environmental context, which I think is what OMI is, but I think maybe you know more about OMI than I do, actually. So.
Yeah, so certainly we use things like gene ontology, PFAM, and KEGG and Metapsych, for example, are annotations that are commonly applied to especially metagenomic data, where we have access to the genomic content of the organisms that comprise a community. And those types of annotation frameworks and ontologies, recognizing that not everything I just mentioned is an ontology per se, provide tremendous interpretability for a good deal of the data that we have access to, because we can put a human readable knowledge-based perspective that drives interpretation of that relationship that we might uncover between, say, a gene family and some health outcome, and at least in the kind of work that we do. But I think another kind of problem that tends to emerge is just whether or not a given gene family or a microbe in question, a taxonomic group, if you will, has had prior evidence of being associated with some sort of ecosystem or covariate of interest in a prior study. And of course, that search space is massive and it would be very difficult to encode. But what OMI is doing, from what I understand, is identifying microorganisms that do associate with different types of locations on the human body that are linked to, say, a given disease, like rheumatoid arthritis, for example. And by encoding information about prior knowledge of observed associations between taxa and different types of covariates, we can go into these knowledge graphs and better ascertain what known information exists about the taxa that we are interested in. I could go on and on about all the caveats and considerations that would have to be baked into this to provide true utility to users, like contextual dependencies, so on and so forth. But I think generally speaking, this is a place where researchers commonly struggle. They make a discovery about a 16S sequence of interest and they don't know how to interpret it. What ends up happening is people start combing through all the prior literature about the taxa of interest that are coming out of their statistical analyses, and it takes a tremendous amount of time, and it is not necessarily comprehensive. So I think this is an area where there may be opportunities to really enrich the advent of discovery in the field.
Sorry, if I could just really quickly on top of that, and that's exactly what I mean. That's exactly what I mean by any data that is not annotated is not useful to the collective, because like he's saying, he's got some gene of interest or some species of interest. Okay, I should be able to go and find any data that has that gene or species or environment of interest like that, and pull it in and pull it into the statistics right away. And if you publish a metagenome and you don't set it up to be done like that, your data is not going to be useful to other people too.
So I think Kai's point also, it kind of leads into the point I was going to respond to, which is that, first of all, I hear you, Thomas. I think you're spot on what we need. But in terms of like someone who has to try to deliver on that, I think technologically what we're going to need to achieve that is a much better ecosystem for distributed resources. Right now, ontologies work really well when you can bring all the data to a central place and build one large knowledge base. We're just now getting to the point where we can start to query across, the different knowledge bases. That notion of linked data is slowly becoming reality. So I think there's a hope that maybe in 5 years we could have that instant answer to query across multiple databases, but we're not quite there yet.
And going back to your previous point, what do you think some of the obstacles are here? What are some of the pain points that could need to be addressed, in order to start building up this knowledge base? Is it better tools for acquisition of that knowledge? Is it making things easier to use, making the ontologies kind of simpler to use? Or is it maybe more, obviously like more funding could help? Or are there kind of other
more funding
parts and sticks that,
But I think actually, Chris, a lot of the stuff that you've been working on, like with LinkML model. So for people in the audience that aren't familiar with it, LinkML is a new a schema language that allows you to build machine readable schemas that can be translated into multiple formats that are readable, but tools that allow us to quickly and easily deliver our knowledge bases and share them with others. It's really, so LinkML is just one piece. So that, exchange formats, query formats that work across different knowledge bases. Those are things that have been the pain points for many years, but I think right now the technology is getting to the point where we can start to apply it. And if we can, but it's still, but getting it to applied at a level where we can scale it really well is not quite there yet.
I could jump in and add to that. I think that in addition to the interface and some of the additional obstacles that Ramona mentioned, there's maybe an awareness issue in the field, and that workshops and events like this can really help to disseminate the gospel, so to speak, not only about what resources exist, but how important it is for people to implement these resources, for example, when they're depositing their data into data repositories. A lot of the microbiome research community, I mean, it's a really interesting field, right? Because you have so many different domains of expertise kind of coming into this newly emergent field. And so they bring with them their cultures and their perspectives. And many of them are conducting this research, not from the perspective of, intimate understanding of data awareness and data standards and best practices, and so to speak, but they're trying to address critical biological questions. And so really disseminating the value of these tools that have been created and the importance of contributing to the growth of the ecosystem of annotated data. If you can build that culture in the research community, I think that's one of the major barriers that we presently face. It's just not something enough people are really really yet participating in. And as we see more and more of this narrative emerge, I have great hope that we'll see more and more people participating in the standards that are being described here.
So I know I already spoke, but I'm going to just chime in real quick with response to that. So one thing I didn't mention during my introduction is that I am a member of the Genomic Standards Consortium. And so I do spend a lot of time trying to working on those mix of standards that both Kai and Chris mentioned. And so I thank you, Tom, for mentioning the need to support standards. I think that what we're lacking in the GSC is funding and resources to go out and really drive education and develop tools for the adoption of those standards. We've worked very hard in the last year to make the standards machine readable, but we need a lot better tooling, and we need people who can go out and develop trainings and share those trainings with the researchers so that they can use. We realize how hard it is to use the standards. We're not doing it just to be obstinate, right? So, but it would be great if we could make them much more user friendly.
Great. And I wonder about, like, the ontologies themselves, the terms in the ontology. Do we have too many ontologies, too few? Do people find the terms that they need in ontologies? And how can do we have sufficient mechanisms in place to kind of engage the biologists to kind of to help develop those ontologies and to help make them, reflect their own kind of conceptual models of their domains?
We have too many and too few terms, right? So when you're a biologist, you go there and you're overwhelmed because there's so many terms, but the one term that you need is not there. That's a classic problem. And that's a case where then so many people just give up because they don't know how that they can go there and in industry, but people just develop their own ontologies if they have the resources rather than contributing their terms back into ontologies. So tools that make it easier. And, I know, again, Chris, you're working on some of these, you've worked on a lot of tools that make it easier for people to contribute back to ontologies, and those are great. But I think there's also a lot of education and outreach that needs to be done to help people understand how to do that.
Mm-hmm, mm-hmm.
Yeah, I would agree with that comment. I think that as the education disseminates some of the confusion about, why there are so many terms, and how the terms that exist have been structured. It's very, it's very well considered. It's very deliberate. They're there for a reason. And then how to fit your own data into the diversity of terms that exist is something that people begin to appreciate the more that they engage the ontologies and understand the underlying frameworks. So I agree. It's the answer is both. But the part of the challenge that we face in this field is that it is moving so rapidly into so many new directions at the same time that you kind of need a lot of terms, because as you begin to explore, say, a particular microbial ecosystem, as we learn more and more, we realize, oh, okay, well, the substructure of the ecosystem may matter. And so we need to start developing ontological terms for characterizing things beyond soil and into, diverse types of soils or, depths within it, so on and so forth. And so it's a complex field that is really integrating a lot of ontological frameworks and has a lot of nuance to it. And the ontologies thus have to encode that nuance to some extent.
So, Tom, I love what you brought up about the soil issue, because that raises an important challenge for ontologists. So all of the three of us here who are ontologists have backgrounds in biology, but we're discovering more and more that in order to describe biological data, we need subject matter experts in soil science, in water, in geology, in social systems, right? So building that interdisciplinary community can be very challenging. That's actually a big part of those two, the iSamples and Sampling Natures grants, though they're not ontology oriented. We need something like that for ontologies as well. And I don't think we have anyone here from the sorry, from the environmental group. Kai, what's the name of the help me out here? Sorry, I'm not going to remember the name, but
I mean, there's
the
groups like
Earth Sciences, ESIP, the Earth Sciences
ESIP.
Information Platform.
Mhm.
You know, so those collaborations between earth scientists and biologists are getting started, and I think that's helpful. But there needs to be more.
Yeah, and for example, with soils, we have like there's a professor at Florida, University of Florida, who's she's wanting to start the soil ontology, and she's, what do I do? Do I go with OBO? Do I build my own thing? She's asking around like, what should I do? Engaging along the line. So I think a lot of community building needs to happen. People who are not familiar with things like OBO, or who are using other systems of ontologies, like it's always a constant battle to try and bring people together. I mean, lots of social challenges too, right? Like when you're contributing to a team project, everyone's personality affects it, and there's lots of different stakeholders with lots of different interests and different timescales and timeframes of when they need or want something done. And so that makes it for another set of logistical challenges. But also a lot of this stuff is unfunded, right? So it's like we kind of do this in our free time to advance the grant while trying to build the longer lasting infrastructure, which is the ontology in the back end, so that hopefully something of that is useful for future grants. But grant funding agencies are like slowly seeming to see this, but maybe not as fast as I would like them to.
And it is fractured, as you say. So ESIP have their own ontology called Suite that is used in some parts of the environmental sciences, which is not generally used for microbiome science. But you've done a lot, Kai, to kind of like help bridge these two communities together and help kind of like work on mappings between Suite and EnvO. Yeah.
Yeah. So efforts like that, so we need to. It's a tricky, people like Chris, he juggles so many projects, right? He contributes to, I don't know, like probably 24 ontologies. He's got like tickets for each one every day. I don't know how he like handles anything. So I think kind of having a better way.
The tickets for other people.
Right. So having kind of a better way to delineate, like what's important to who, when, and like how does it maximize the benefit to as many people as possible, while kind of accepting that you're not going to get everything.
Okay, we should let's switch over to some questions from the attendees. So we've got some in the Google Doc here right now. So maybe let's just start with the first one. So how do we get around walled gardens, these different databases, IMG, Biosite, KEGG, and get a Rosetta Stone of identifiers? So I hope I did that question justice there. Does anyone want to address that?
Chris, maybe you can just briefly explain like how you made NCBI taxonomy into an ontology, and like how that's kind of an example of how you did that process, or do you have a better example you want to respond with?
In some ways, that's too straightforward. It's basically a Python script.
Right, but the idea that you took like an existing database and you turned it into a common format that is accessible in an ontology way. I'm not sure.
Yeah. So a lot of it is really just kind of adopting the OBO principles and kind of like making sure that identifiers are also kind of like resolvable URLs, that we use a kind of like a standard data model for being able to say that this is the primary name of this taxon. These are its synonyms. This is its kind of parent taxon. These are our child taxon. So I think, just rather than developing kind of bespoke systems, is really to just kind of like adopt some of the existing standards, for ontologies, using kind of formats like OBO and OWL, and using some of the standard ways of providing metadata in ontologies go a long way to making a lot of these different kind of like walled gardens, kind of interoperable with one another.
So we should just like make it KEGG, et cetera, ontology kind of.
Well, I'd actually like to advocate a little bit against that, because I think that, even though I use the NCBI taxon all the time and I see its value, it's better to have that ontology maintained by the source who's maintained. It would be better to have each of these sources maintained, not necessarily their own ontology. These could just, but to have, resolvable URLs for each of their terms. And if there's a hierarchy, to implement that hierarchy. So it doesn't have to be a very deep, a lot of Sorry, it's very, very loud right now. So I'm going to mute and let someone else talk for a minute.
Yeah, sorry, that was what I meant to say, like taking the existing KEGG and COG and those things and just kind of making its terms resolve within the same framework as these other ontologies. I think that's what we were going at.
Yeah, so again, getting back to like, this notion of linked data, right? So can we work with the resources who are unintentionally, I think, putting up walls to help them bring those walls down so that we don't have to reinvent them, so that we can, again, get back to that concept of interoperability and distributed data and federation that I was mentioning earlier. But yes, they all need to have, first step, they all need to have globally unique, permanent, resolvable identifiers for all of their concepts. That would be a really great help. And then to add in, as Chris was mentioning, the metadata that describes, have a definition, have synonyms, et cetera, additional metadata with each term.
Okay. So another sort of like quick practical question here is like someone is saying they've fallen down the ontology rabbit hole, which I think many of us are familiar with, working with human data using the ENA browsers. Are there better ways to access this data to walk these ontology trees? What are people's general approaches here?
I'll just throw it in the
Go ahead.
I'll just throw it in the chat. There's a really good tool by the European Bioinformatics Institute, which is a lookup browser. I just put it in the chat. I think that's Chris and I's preferred way of doing this, but I'm happy to hear what others have to say.
I love to use it. OLS is a great tool. As a developer, I use OntoB sometimes, but for non-ontologists, that one might be a little more challenging. And of course, BioPortal. There's lots of places, but the challenge is I think is when you don't know what ontologies to look at and what branches. And so that's why I really love that you're doing for NMDC is that you're making subsets of ontologies that can really focus. And I think that's what we need is communities that work together, like domain-specific communities that work together with the ontology and knowledge management and standards communities to build the toolings that work for their community. We can't build one. If we were to build an all-purpose ontology server, we end up with something like OLS. It's not even all-purpose. It's just within sort of life sciences, health sciences. So we need to have tools like OLS that are specific to specific domains. So could there be an NMDC vocabulary server, an NMDC version of OLS that just serves the ontologies and subsets of ontologies that metagenomics researchers need, for example?
Great. Yeah. No, I think that's key. I mean, OLS is a wonderful tool, but it will just show you the ontology itself. It doesn't show you the data annotated with the ontology, which I think is what many people really need to be seeing.
Right. And so like within BICO-DEMO, for example, we're making these curated subsets similar to what you're doing with NMDC, and we're just showing that to our data managers who then have a smaller set of things to look at to make their annotations.
Mm-hmm. Right. All right. So just one more question to kind of close off, returning to the kind of like, the carrot stick discussion. So what are some of the kind of professional benefits, both for early career researchers and more kind of established ones, for contributing to an ontology versus just getting the answers you need for your own kind of like research and tool development and not contributing back? How can we better enable people to encourage people to contribute?
I can jump in with an initial answer.
Yeah, please.
I don't know that it's the best answer, but I think it's an important one. And it's so much of what we do in science depends upon the community. And we're capitalizing upon community-driven infrastructure, resources, expertise, investment. And we should be looking at ontologies as being akin to so much of the infrastructure that we tend to rely upon in other aspects of our research. And what we do when we contribute to ontologies is we are helping to build out a future infrastructure that can pay dividends on our time. If we make our data and our results consistently machine readable, then it means that going forward, we have improved opportunities to rapidly access mine and interpret those data and results. And we benefit from the collective contributions of data and results across our community. So I don't know that it's necessarily the type of activity that provides an instantaneous short-term return on investment, but it is something that helps to secure your ability to innovate as you go forward over the course of your career. And thus, I think it's worthwhile.
I'll chime in with the same argument that's used for why you should have a good data management plan. The person that's most likely to reuse your data is you. And so managing your data well, making it as useful as possible is it will have the benefit then of helping others to reuse your data. But there's a good chance that you're going to want to reuse your own data in the future. So that's a nice sort of selfish way of looking at it.
As for a practical, for early stage career people, like for my own experiences, it's been difficult, but at the same time, I've had lots of opportunities to hop from job to job to job, solving the same problem over and over again for different people, because, there are very few people who are actually helping to contribute to what that specific thing at that moment that someone needs. So I don't know, it's not going to get you through PhD. Like my committee was not particularly interested in ontology development. I'll put it that way.
All right.
But that said, if you become an ontologist, there are lots of jobs out there. Like probably like, I'm always trying to hire a good ontologist.
Yeah, talk to us first, please.
Right. So there is a career path out there for it.
Right. Okay. On that note, we should probably wrap up. So thank you so much to Tom, Ramona, and Kai. That was a great panel, I thought. And special thanks to Marissa as well, who helped put all of this together. And thanks to all of you for attending. This was recorded. It will be posted on our website. There's a link in the notes. There's a December webinar that I encourage you to register for as well. We'll have a slide now. Oh, do we have a slide for that? I don't know if you want to show that, Marissa, but the link should be on the notes doc that you all have as well. And also, please join our community Slack channel as well. You can find the link to that there. And sorry we didn't get to all of your questions, but please just join the Slack. And many of us are there, and we can continue the conversation there. And so with that, just thanks again. I thought that was great, and I'll see you all later on.
Thanks a lot.